So our next speaker is Dr. Giulia Gallo from the Pirbright Institute, and she's going to talk about the insight into the scope of alphacoronavirus receptor usage.
Okay, so thank you for the introduction. So I'm going to talk about the spikes and receptor usage. So the spike of alphacoronaviruses is organized in two domains, the S1, which is the attachment protein, and S2, which is the fusion protein. So if we have a look on the viral envelope, this is how the spike looks like, is a trimer. And if we color code it, what you see in red is the fusion protein, in yellow is the attachment protein. And as you can see, at the apical part of the spike, we have a bluish domain, which is the RBD, which is included in the attachment protein. And the RBD is the receptor binding domain. In the case of most of alphacoronaviruses, this receptor binding domain contacts the non-receptor, which is the aminopeptidase N, and this is how it looks like. If we look in more detail, we can see that the RBD of alphacoronaviruses interact with the APN through 3 different loops. And what's interesting is that although different virus species of alphacoronavirus use APN, they do not interact with the same region of APN. So here I'm showing you the top view of APN, and highlighted are 2 footprints, one of canine coronavirus and the other of human coronavirus. And you can see that they don't even overlap. So this is where our work started, and the goal of our study was to identify how many alphacoronaviruses use APN for entry, and after defining those, to see how many animal species are permissive to APN use in alphacoronaviruses, and in even greater detail, what are the residues that determine host range, APN-related host range. And this goes in a bigger question that we have in the lab, which is, can we predict host range based on the attachment proteins of viruses and the proteinaceous receptor they use for entry? So the first category of stores are spikes. So we used an algorithm to select in an unbiased way 40 different spikes. Here you can see them listed. And alphacoronaviruses are hosted by bats, so the majority of them was actually from bats. Then we have the human coronavirus, which is endemic in the population, Tutunae, and few domesticated animals, among which canine coronaviruses, feline and porcine coronaviruses. So we took this spike, we synthesized them, and then we used pseudotyping for our entry assay. So the first thing we had done was to check that the spike could be properly pseudotyped. So we just used pseudotyping system, purified the particles, and ran a Western blot to see that the spike was incorporated. And we were quite lucky because most of them were successful and we could see the spike. And in parallel, we also built a library of receptor of APN from different animal species, namely carnivores, ruminants, and a lot of bats. And we transited transfected cells and checked by flow cytometry that the receptor was expressed, and we were also successful in that. So after we had these 2 matrices of spike and APN, the next step was to mix all of them and see, and that's what we've done with this big matrix of screening. And this is the main result we got. So where you see a cross is that we didn't see pseudotyping. When you see a gray square, it means that that spike could not enter with any of the APN we tested. And finally, the square in purple means that that virus could use at least 1 APN. And if we go in further detail, we then characterize which animal and how many animals could support entry. And based on those, we classified alphacoronavirus in 3 groups. So the multi-order specialists, they use just a few APN from different mammalian orders. Then an order generalist, they uses a lot of different APN from the same mammalian order. And finally, the 2 specialists, they use just few APN from just 1 mammalian order. And what I'm going to talk next is just two scenarios that I'm going to detail more. The first scenario is the case of Tutunae. So as you know, Tutunae is an endemic human virus, but it is believed that it's derived from bats. And actually, when we look in bat population, 229-like viruses have been found. So our algorithm selected the spike of one bat 229, and we assessed the host range. We first discovered that it could also use APN, and the host range was quite narrow. Then, for the story of 229, it is believed that at some point it spilled over to probably an intermediate host, went to cycle of recombination adaptation, and finally spilled again to human, where it went to probably lost its NTD and also got adapted to the human population. Our algorithm also selected a human 229. We assessed the host range, and as you can see, it's quite similar, with the exception that the human virus uses human APN. So both this highlights the 229 are multi-order specialists. Then we went in further detail and we compared the sequences of the spike, in particular the RBD, and we found that at position 316 there was an interest substitution between human and bat 229. So for human we have a lysine, for bat we have a serine, which you will know they have quite different biochemical properties. So I made a point mutation. This, you can see the position. You can see it's at the interface of interaction with human APN. So I made a point mutant at introducing the bat seri in the human spike. And as you can see, just this point mutant could confer broader host range, especially for the usage of bat APNs. Then you might know that some coronaviruses can be primed, meaning that a cellular protease can come and cleave the fusion protein, so to expose the fusion peptide and facilitate entry. And because this is done by cellular protease, we repeat our screening, but not just in presence of APNs, but also in presence of TMPRSS2. And as you can see from the result, this had a limited impact for human 229, but it had a bigger impact for bat 229, where the priming increases the host range of the virus. So the second case study I want to show you concerns canine coronaviruses. So our algorithm selected two canine coronaviruses that you can see here in red, A76 and SDF3, but we also included two canine coronavirus with pandemic potential that you can see in orange, Z19 and UPN18. And you can already see by this that the host range of these viruses is quite different. We then compared the similarity of the RBD and the spike, and I decided to focus on the two most similar generalist and specialist. For the specialist, we took SDF3, and for the generalist, UPN18. And we analyzed the RBD. And what I've done was to swap the RBDs. So I introduced the RBD of the specialist into the spike of the generalist and vice versa, hoping to restore generalism and specialism. But you can see that the story is not this straight, because when we introduce the RBD of the specialist into the spike of the generalist, we could not restore specialism and vice versa. So this means that the RBD on its own for canine coronavirus does not explain the host range that we observe. And the story is not over, it's even more complex, because as you might know, we have an alphacoronavirus which infects human that doesn't use APN but uses ACE2. And I was quite lucky in the lab because we already had a library of ACE2 from different animal species, so I pseudotype NL63 and just assess the host range. And as you can see, NL63 is quite a generalist. And so then the next step was to compare the sequences of permissive versus non-permissive ACE2. And I found that at the position 354 of ACE2, we have a glycine for all the permissive species, but we have a polar residue for everything that's not permissive. And this is how it looks like. This residue is at the interface of interaction with the RBD of NL63. So again, I did point mutant where I introduced this glycine into non-permissive species, and I could restore permissivity, meaning that this residue is really important for NL63 host range. So the bigger question I thought I discussed at the beginning, can we predict host range based on amino acid sequence and receptors? The answer is it's not that easy, because host range depends on the proteinaceous receptor, but as I show you for Batu2-E, it also depends on cofactor and co-receptors. It depends on the sequence of the RBD, as shown by human 2-E, and how we could modify host range based on one point mutation. But then the canine coronavirus story showed us that it's not just the RBD, but in some cases it's also other domain like the fusion protein or the NTD of the spike. So moving forward, how we can determine generalism and specialism? Well, we will need much more data. And this could come from the discovery of new receptor, from analyzing a host restriction factor, to study the specific contribution of individual amino acid to host range, and finally with study of in vitro evolution. And we can imagine that we could put together all this data in a big data set that could account for the host range based on the different steps of the viral cycle. And finally, we could feed this to an algorithm that could help us identify features that might be important for host range. But I'm leaving you to this. This might have ethical implications that should maybe be discussed before starting such study. And the most important slide, I want to thank you all the people that supported me and the groups where I work in, because they make science possible and enjoyable even when you get a big matrix of negative data. So my group at Perbright and the group I'm working in doing structural study at Cambridge. And thank you for your attention.
Should work.
Thanks for a great talk. I was quite struck by your 63 single amino acid chain. I was just wondering, how reflective of actual infection do you think your model is, and do you have an eye towards validating that through any kind of reverse genetics approach?
So I've been talking to someone, and my understanding is that I don't do like live virus work. My understanding is that it's for an NS63. For example, if you want to try in vivo, it's quite hard. There is not a really good system, so that would be quite challenging. I don't work with live viruses, so I think there would be a way, because we could do a similar screening with the different ACE2 just using the live virus. So that's something that could be done. But unfortunately, we see a restriction for New World monkeys. If it doesn't work, we will need to create like transgenic non-human primate, and that's quite challenging. Yeah.
Can try, but it's not working.
Thanks. Great talk. I was just wondering, actually, so I do know that ACE2 or NS3 and SARS-CoV-2 bind to different regions of ACE.
Yeah.
But I was wondering if this 3x4 residue has any functional relevance for SARS-CoV-2 infection.
Yes. So if you have a look at the footprint of ACE2 where SARS-2 and NS3 bind, you will see that the residue 354, it's involved in the binding of both. But for NS63, the glycine is really in the middle of the footprint, while for SARS-2 is at the extremity. I think that even for SARS-2, there has been shown that it's limited relevance, but I don't know. Because of the position, I reckon there might be a bigger role for NS63 than for SARS-2.
Thank you. Well, we'll
Yeah, if we don't have any more questions, we can go straight to the last talk before the flash talks.